Hemoglobin, American, or hemoglobin, British, /Hemlobin, H-mo/, abbreviated HB or hemoglobin, is the iron-containing oxygen transport metalloprotein in the red blood cells of all vertebrates, with the exception of the fish family Channicthiidae, as well as the tissues of some invertebrates. Hemoglobin in the blood carries oxygen from the respiratory organs, lungs or gills, to the rest of the body, i.e. the tissues. There it releases the oxygen to permit aerobic respiration to provide energy to power the functions of the organism in the process called metabolism. In mammals, the protein makes up about 96% of the red blood cells' dry content, by weight, and around 35% of the total content, including water. Hemoglobin has an oxygen binding capacity of 1.34 mLO2 per gram, which increases the total blood oxygen capacity 70-fold compared to dissolved oxygen in blood. The mammalian hemoglobin molecule can carry, up to 4 oxygen molecules. Hemoglobin is involved in the transport of other gases. It carries some of the body's respiratory carbon dioxide, about 20–25% of the total, as carbaminohemoglobin, in which CO2 is bound to the globin protein. The molecule also carries the important regulatory molecule nitric oxide bound to a globin protein thiol group, releasing it at the same time as oxygen. Hemoglobin is also found outside red blood cells and their progenitor lines. Other cells that contain hemoglobin include the A9 dopaminergic neurons in the substantia nigra, macrophages, alveolar cells, and mesangial cells in the kidney. In these tissues, hemoglobin has a non-oxygen carrying function as an antioxidant and a regulator of iron metabolism. Hemoglobin and hemoglobin-like molecules are also found in many invertebrates, fungi, and plants. In these organisms, hemoglobins may carry oxygen, or they may act to transport and regulate other small molecules and ions such as carbon dioxide, nitric oxide, hydrogen sulfide, and sulfide. A variant of the molecule, called leg hemoglobin, is used to scavenge oxygen away from anaerobic systems, such as the nitrogen-fixing nodules of leguminous plants, before the oxygen can deactivate, the system. Research history. In 1825 J. F. Engelhard discovered that the ratio of Fe to protein is identical in the hemoglobins of several species. From the known atomic mass of iron he calculated the molecular mass of hemoglobin to N times 16,000, N equals number of iron atoms per hemoglobin, now known to be 4, the first determination of a protein's molecular mass. This hasty conclusion drew a lot of ridicule at the time from scientists who could not believe that any molecule could be that big. Gilbert Smithson Adair confirmed Engelhard's results in 1925 by measuring the osmotic pressure of hemoglobin solutions. The oxygen-carrying protein hemoglobin was discovered by Hunefeld in 1840. In 1851, German physiologist Otto Funk published a series of articles in which he described growing hemoglobin crystals by successively diluting red blood cells with a solvent such as pure water, alcohol, or ether, followed by slow evaporation of the solvent from the resulting protein solution. Hemoglobin's reversible oxygenation was described a few years later by Felix Hoppe Seiler. In 1959, Max Perutz determined the molecular structure of hemoglobin by X-ray crystallography. This work resulted in his sharing with John Kendrew the 1962 Nobel Prize in Chemistry for their studies of the structures of globular proteins. The role of hemoglobin in the blood was elucidated by French physiologist Claude Bernard. The name hemoglobin is derived from the words heme and globin, reflecting the fact that each subunit of hemoglobin is a globular protein with an embedded heme group. Each heme group contains one iron atom, that can bind one oxygen molecule through ion-induced dipole forces. The most common type of hemoglobin in mammals contains 4 such subunits. Genetics. Hemoglobin consists of protein subunits, the globin molecules, and these proteins, in turn, are folded chains of a large number of different amino acids called polypeptides. The amino acid sequence of any polypeptide created by a cell is in turn determined by the stretches of DNA called genes. In all proteins, it is the amino acid sequence that determines the protein's chemical properties and function. There is more than one hemoglobin gene. In humans, hemoglobin A, the main form of hemoglobin present, is coded for by the genes HbA1, HbA2, and HbB. The amino acid sequences of the globin proteins in hemoglobins usually differ between species. These differences grow with evolutionary distance between species. For example, the most common hemoglobin sequences in humans and chimpanzees are nearly identical, differing by only one amino acid in both the alpha and the beta globin protein chains. These differences grow larger between less closely related species. Even within a species, different variants of hemoglobin always exist, although one sequence is usually a most common one in each species. Mutations in the genes for the hemoglobin protein in a species result in hemoglobin variants. Many of these mutant forms of hemoglobin cause no disease. Some of these mutant forms of hemoglobin, however, cause a group of hereditary diseases termed the hemoglobinopathies. The best-known hemoglobinopathy is sickle cell disease, which was the first human disease whose mechanism was understood at the molecular level. A mostly separate set of diseases called thalassemias involves underproduction of normal and sometimes abnormal hemoglobins through problems and mutations in globin gene regulation. All these diseases produce anemia. Variations in hemoglobin amino acid sequences, as with other proteins, may be adaptive. For example, hemoglobin has been found to adapt in different ways to high altitudes. Organisms living at high elevations experience lower partial pressures of oxygen compared to those at sea level. This presents a challenge to the organisms that inhabit such environments because hemoglobin, which normally binds oxygen at high partial pressures of oxygen, must be able to bind oxygen when it is present at a lower pressure. Different organisms have adapted to such a challenge. For example, recent studies have suggested genetic variants in deer mice that help explain how deer mice that live in the mountains are able to survive in the thin air that accompanies high altitudes. A researcher from the University of Nebraska-Lincoln found mutations in 4 different genes that can account for differences between deer mice that live in lowland prairies versus the mountains. After examining wild mice captured from both highlands and lowlands, it was found that the genes of the 2 breeds are virtually identical except for those that govern the oxygen-carrying capacity of their hemoglobin. The genetic difference enables highland mice to make more efficient use of their oxygen, since less is available at higher altitudes, such as those in the mountains. Mammoth hemoglobin featured mutations that allowed for oxygen delivery at lower temperatures, thus enabling mammoths to migrate to higher latitudes during the Pleistocene. This was also found in hummingbirds that inhabit the Andes. Hummingbirds already expend a lot of energy and thus have high oxygen demands, and yet Andean hummingbirds have been found to thrive in high altitudes. Non-synonymous mutations in the hemoglobin gene of multiple species living at high elevations, Oreotrochilus, A. castelnaudii, C. violifera, P. gigas, and A. viridicuata, have caused the protein to have less of an affinity for inositol hexaphosphate, IHP, a molecule found in birds that has a similar role as 2,3-BPG in humans. This results in the ability to bind oxygen in lower partial pressures. It should be noted, however, that birds' unique circulatory lungs also promote efficient use of oxygen at low partial pressures of O2. These 2 adaptations reinforce each other and account for birds' remarkable high-altitude performance. Hemoglobin adaptation extends to humans, as well. Studies have found that a small number of native Tibetan women have a genotype which codes for hemoglobin to be more highly saturated with oxygen. Natural selection seems to be the main force working on this gene because the mortality rate of offspring is significantly lower for women with higher hemoglobin-oxygen affinity when compared to the mortality rate of offspring from women with low hemoglobin-oxygen affinity. While the exact genotype and mechanism by which this occurs is not yet clear, selection is acting on these women's ability to bind oxygen in low partial pressures, which overall allows them to better sustain crucial metabolic processes. Synthesis. Hemoglobin, Hb, is synthesized in a complex series of steps. The heme part is synthesized in a series of steps in the mitochondria and the cytosol of immature red blood cells, while the globin protein parts are synthesized by ribosomes in the cytosol. Production of Hb continues in the cell throughout its early development from the proerythroblast to the reticulocyte in the bone marrow. At this point, the nucleus is lost in mammalian red blood cells, but not in birds and many other species. Even after the loss of the nucleus in mammals, residual ribosomal RNA allows further synthesis of Hb until the reticulocyte loses its RNA soon after entering the vasculature. This hemoglobin synthetic RNA in fact gives the reticulocyte its reticulated appearance and name. Structure. Hemoglobin has a quaternary structure characteristic of many multi-subunit globular proteins. Most of the amino acids in hemoglobin form alpha helices, and these helices are connected by short non-helical segments. Hydrogen bonds stabilize the helical sections inside this protein, causing attractions within the molecule, which then causes each polypeptide chain to fold into a specific shape. Hemoglobin's quaternary structure comes from its 4 subunits in roughly a tetrahedral arrangement. In most vertebrates, the hemoglobin molecule is an assembly of 4 globular protein subunits. Each subunit is composed of a protein chain tightly associated with a non-protein prosthetic heme group. Each protein chain arranges into a set of alpha helix structural segments connected together in a globin fold arrangement. Such a name is given because this arrangement is the same folding motif used in other heme/globin proteins such as myoglobin. This folding pattern contains a pocket that strongly binds the heme group. A heme group consists of an iron, Fe, ion, charged atom, held in a heterocyclic ring, known as a porphyrin. This porphyrin ring consists of 4 pyrrole molecules cyclically linked together, by methine bridges, with the iron ion bound in the center. The iron ion, which is the site of oxygen binding, coordinates with the 4 nitrogen atoms in the center of the ring, which all lie in one plane. The iron is bound strongly, covalently, to the globular protein via the N atoms of the imidazole ring of F8 histidine residue, also known as the proximal histidine, below the porphyrin ring. A 6th position can reversibly bind oxygen by a coordinate covalent bond, completing the octahedral group of 6 ligands. Oxygen binds in an end-on bent geometry where one oxygen atom binds to Fe and the other protrudes at an angle. When oxygen is not bound, a very weakly bonded water molecule fills the site, forming a distorted octahedron. Even though carbon dioxide is carried by hemoglobin, it does not compete with oxygen for the iron binding positions but is bound to the protein chains of the structure. The iron ion may be either in the Fe2+ or in the Fe3+ state, but ferrihemoglobin, methemoglobin, Fe3+, cannot bind oxygen. In binding, oxygen temporarily and reversibly oxidizes, Fe2+, to Fe3+, while oxygen temporarily turns into the superoxide ion, thus iron must exist in the +2 oxidation state to bind oxygen. If superoxide ion associated to Fe3+ is protonate, the hemoglobin iron will remain oxidized and incapable of binding oxygen. In such cases, the enzyme methemoglobin reductase will be able to eventually reactivate methemoglobin by reducing the iron center. In adult humans, the most common hemoglobin type is a tetramer, which contains 4 subunit proteins, called hemoglobin A, consisting of 2 alpha and 2 beta subunits non-covalently bound, each made of 141 and 146 amino acid residues, respectively. This is denoted as alpha-2 beta-2. The subunits are structurally similar and about the same size. Each subunit has a molecular weight of about 16,000 daltons, for a total molecular weight of the tetramer of about 64,000 daltons, 64,458 g/mol. Thus, 1 g/dl equals 0.1551 mol/L. Hemoglobin A is the most intensively studied of the hemoglobin molecules. In human infants, the hemoglobin molecule is made up of 2 alpha chains and 2 gamma chains. The gamma chains are gradually replaced by beta chains as the infant grows. The 4 polypeptide chains are bound to each other by salt bridges, hydrogen bonds, and the hydrophobic effect. Oxygen saturation. In general, hemoglobin can be saturated with oxygen molecules, oxyhemoglobin, or desaturated with oxygen molecules, deoxyhemoglobin. Oxyhemoglobin. Oxyhemoglobin is formed during physiological respiration when oxygen binds to the heme component of the protein hemoglobin in red blood cells. This process occurs in the pulmonary capillaries adjacent to the alveoli of the lungs. The oxygen then travels through the bloodstream to be dropped off at cells where it is utilized as a terminal electron acceptor in the production of ADP by the process of oxidative phosphorylation. It does not, however, help to counteract a decrease in blood pH. Ventilation, or breathing, may reverse this condition by removal of carbon dioxide, thus causing a shift up in pH. Hemoglobin exists in 2 forms, a taut, tense form, T, and a relaxed form, R. Various factors such as low pH, high CO2 and high 2,3 bpg at the level of the tissues favor the taut form, which has low oxygen affinity and releases oxygen in the tissues. Conversely, a high pH, low CO2, or low 2,3 bpg favors the relaxed form, which can better bind oxygen. The partial pressure of the system also affects O2 affinity where, at high partial pressures of oxygen, such as those present in the alveoli, the relaxed, high affinity, R, state is favored. Inversely, at low partial pressures, such as those present in respiring tissues, the low affinity, T, tense state is favored. Additionally, the binding of oxygen to the iron, 2, heme pulls the iron into the plane of the porphyrin ring, causing a slight conformational shift. The shift encourages oxygen to bind to the 3 remaining heme units within hemoglobin, thus, oxygen binding is cooperative. Deoxygenated hemoglobin. Deoxygenated hemoglobin is the form of hemoglobin without the bound oxygen. The absorption spectra of oxyhemoglobin and deoxyhemoglobin differ. The oxyhemoglobin has significantly lower absorption of the 660 nm wavelength than deoxyhemoglobin, while at 940 nm its absorption is slightly higher. This difference is used for the measurement of the amount of oxygen in a patient's blood by an instrument called a pulse oximeter. This difference also accounts for the presentation of cyanosis, the blue to purplish color that tissues develop during hypoxia. Deoxygenated hemoglobin is paramagnetic, it is weakly attracted to magnetic fields. In contrast, oxygenated hemoglobin exhibits diamagnetism, a weak repulsion from a magnetic field. Evolution of vertebrate hemoglobin. Scientists agree that the event that separated myoglobin from hemoglobin occurred after lampreys diverged from jawed vertebrates. This separation of myoglobin and hemoglobin allowed for the different functions of the 2 molecules to arise and develop. Myoglobin has more to do with oxygen storage while hemoglobin is tasked with oxygen transport. The alpha and beta-like globin genes encode the individual subunits of the protein. The predecessors of these genes arose through another duplication event also after the Nat-Hosum common ancestor derived from jawless fish, approximately 450–500 million years ago. The development of alpha and beta genes created the potential for hemoglobin to be composed of multiple subunits, a physical composition central to hemoglobin's ability to transport oxygen. Having multiple subunits contributes to hemoglobin's ability to bind oxygen cooperatively as well as be regulated allosterically. Subsequently, the alpha gene also underwent a duplication event to form the HbA1 and HbA2 genes. These further duplications and divergences have created a diverse range of alpha and beta-like globin genes that are regulated so that certain forms occur at different stages of development. Most ice fish of the family Channichthyeidae have lost their hemoglobin genes as an adaptation to cold water. Iron's oxidation state in oxyhemoglobin. Assigning oxygenated hemoglobin's oxidation state is difficult because oxyhemoglobin, HbO2, by experimental measurement, is diamagnetic, no net unpaired electrons, yet the lowest energy, ground state, electron configurations in both oxygen and iron are paramagnetic, suggesting at least one unpaired electron in the complex. The lowest energy form of oxygen, and the lowest energy forms of the relevant oxidation states of iron, are these. Triplet oxygen, the lowest energy molecular oxygen species, has two unpaired electrons in antibonding pi* molecular orbitals. Iron, 2, tends to exist in a high-spin 3d6 configuration with 4 unpaired electrons. Iron, 3, 3d5, has an odd number of electrons, and thus must have one or more unpaired electrons, in any energy state. All of these structures are paramagnetic, have unpaired electrons, not diamagnetic. Thus, a non-intuitive, e.g., a higher energy for at least one species, distribution of electrons in the combination of iron and oxygen must exist, in order to explain the observed diamagnetism and no unpaired electrons. The two logical possibilities to produce diamagnetic, no net spin, HbO2 are: low spin Fe2+ binds to singlet oxygen. Both low spin iron and singlet oxygen are diamagnetic. However, the singlet form of oxygen is the higher energy form of the molecule. Low spin Fe3+ binds to O2, the superoxide ion, and the two unpaired electrons couple antiferromagnetically, giving observed diamagnetic properties. Here, the iron has been oxidized, has lost one electron, and the oxygen has been reduced, has gained one electron. Another possible model in which low spin Fe4+ binds to peroxide, O22, can be ruled out by itself, because the iron is paramagnetic, although the peroxide ion is diamagnetic. Here, the iron has been oxidized by 2 electrons, and the oxygen reduced by 2 electrons. Direct experimental data. X-ray photoelectron spectroscopy suggests iron has an oxidation state of approximately 3.2. Infrared vibrational frequencies of the OO bond suggests a bond length fitting with superoxide, a bond order of about 1.6, with superoxide being 1.5. X-ray absorption near edge structures at the iron K edge. The energy shift of 5 eV between deoxyhemoglobin and oxyhemoglobin, as for all the methemoglobin species, strongly suggests an actual local charge closer to Fe3+ than Fe2+. Thus, the nearest formal oxidation state of iron in HbO2 is the +3 state, with oxygen in the 1 state, as superoxide. O2. The diamagnetism in this configuration arises from the single unpaired electron on superoxide aligning antiferromagnetically with the single unpaired electron on iron, in a low spin d5 state, to give no net spin to the entire configuration, in accordance with diamagnetic oxyhemoglobin from experiment. The second choice of the logical possibilities above for diamagnetic oxyhemoglobin being found correct by experiment, is not surprising. Singlet oxygen, possibility number 1, is an unrealistically high energy state. Model 3 leads to unfavorable separation of charge, and does not agree with the magnetic data, although it could make a minor contribution as a resonance form. Iron's shift to a higher oxidation state in HBO2 decreases the atom's size, and allows it into the plane of the porphyrin ring, pulling on the coordinated histidine residue and initiating the allosteric changes seen in the globulins. Early postulates by bioinorganic chemists claimed that possibility number 1, above, was correct and that iron should exist in oxidation state 2. This conclusion seemed likely, since the iron oxidation state 3 is methemoglobin, when not accompanied by superoxide. O2 to hold the oxidation electron, was known to render hemoglobin incapable of binding normal triplet O2 as it occurs in the air. It was thus assumed that iron remained as Fe2, when oxygen gas was bound in the lungs. The iron chemistry in this previous classical model was elegant, but the required presence of the diamagnetic, high-energy, singlet oxygen molecule was never explained. It was classically argued that the binding of an oxygen molecule placed high-spin iron, 2, in an octahedral field of strong field ligands. This change in field would increase the crystal field splitting energy, causing iron's electrons to pair into the low-spin configuration, which would be diamagnetic in Fe2. This forced low-spin pairing is indeed thought to happen in iron when oxygen binds, but is not enough to explain iron's change in size. Extraction of an additional electron from iron by oxygen is required to explain both iron's smaller size and observed increased oxidation state, and oxygen's weaker bond. The assignment of a whole number oxidation state is a formalism, as the covalent bonds are not required to have perfect bond orders involving whole electron transfer. Thus, all three models for paramagnetic HBO2 may contribute to some small degree, by resonance, to the actual electronic configuration of HBO2. However, the model of iron in HBO2 being Fe3, is more correct than the classical idea that it remains Fe2. Cooperativity. When oxygen binds to the iron complex, it causes the iron atom to move back toward the center of the plane of the porphyrin ring, see moving diagram. At the same time, the imidazole side chain of the histidine residue interacting at the other pole of the iron is pulled toward the porphyrin ring. This interaction forces the plane of the ring sideways toward the outside of the tetramer, and also induces a strain in the protein helix containing the histidine as it moves nearer to the iron atom. This strain is transmitted to the remaining three monomers in the tetramer, where it induces a similar conformational change in the other heme sites such that binding of oxygen to these sites becomes easier. As oxygen binds to one monomer of hemoglobin, the tetramer's conformation shifts from the T, tense, state to the R, relaxed, state. This shift promotes the binding of oxygen to the remaining three monomers heme groups, thus saturating the hemoglobin molecule with oxygen. In the tetrameric form of normal adult hemoglobin, the binding of oxygen is, thus, a cooperative process. The binding affinity of hemoglobin for oxygen is increased by the oxygen saturation of the molecule, with the first molecules of oxygen bound influencing the shape of the binding sites for the next ones, in a way favorable for binding. This positive cooperative binding is achieved through steric conformational changes of the hemoglobin protein complex as discussed above, i.e., when one subunit protein in hemoglobin becomes oxygenated, a conformational or structural change in the whole complex is initiated, causing the other subunits to gain an increased affinity for oxygen. As a consequence, the oxygen binding curve of hemoglobin is sigmoidal, or S-shaped, as opposed to the normal hyperbolic curve associated with non-cooperative binding. The dynamic mechanism of the cooperativity in hemoglobin and its relation with the low-frequency resonance has been discussed. Binding for ligands other than oxygen. Besides the oxygen ligand, which binds to hemoglobin in a cooperative manner, Hemoglobin ligands also include competitive inhibitors such as carbon monoxide, CO, and allosteric ligands such as carbon dioxide, CO2, and nitric oxide, NO. The carbon dioxide is bound to amino groups of the globin proteins to form carbaminohemoglobin. This mechanism is thought to account for about 10% of carbon dioxide transport in mammals. Nitric oxide can also be transported by hemoglobin. It is bound to specific thiol groups in the globin protein to form an S-nitrosithiol, which dissociates into free nitric oxide and thiol again, as the hemoglobin releases oxygen from its heme site. This nitric oxide transport to peripheral tissues is hypothesized to assist oxygen transport in tissues, by releasing vasodilatory nitric oxide to tissues in which oxygen levels are low. Competitive. The binding of oxygen is affected by molecules such as carbon monoxide, for example, from tobacco smoking, exhaust gas, and incomplete combustion in furnaces. CO competes with oxygen at the heme binding site. Hemoglobin's binding affinity for CO is 250 times greater than its affinity for oxygen, meaning that small amounts of CO dramatically reduce hemoglobin's ability to transport oxygen. Since carbon monoxide is a colorless, odorless, and tasteless gas, and poses a potentially fatal threat, carbon monoxide detectors have become commercially available to warn of dangerous levels in residences. When hemoglobin combines with CO, it forms a very bright red compound called carboxyhemoglobin, which may cause the skin of CO poisoning victims to appear pink in death, instead of white or blue. When inspired air contains CO levels as low as 0.02%, headache and nausea occur. If the CO concentration is increased to 0.1%, unconsciousness will follow. In heavy smokers, up to 20% of the oxygen-active sites can be blocked by CO. In similar fashion, hemoglobin also has competitive binding affinity for cyanide, CN, sulfur monoxide, SO, and sulfide, S2, including hydrogen sulfide, H2S. All of these bind to iron in heme without changing its oxidation state, but they nevertheless inhibit oxygen binding, causing grave toxicity. The iron atom in the heme group must initially be in the ferrous, Fe2+, oxidation state to support oxygen and other gases binding and transport. It temporarily switches to ferric during the time oxygen is bound, as explained above. Initial oxidation to the ferric, Fe3+, state without oxygen converts hemoglobin into hemoglobin or methemoglobin, which cannot bind oxygen. Hemoglobin in normal red blood cells is protected by a reduction system to keep this from happening. Nitric oxide is capable of converting a small fraction of hemoglobin to methemoglobin in red blood cells. The latter reaction is a remnant activity of the more ancient nitric oxide dioxygenase function of globins. Allosteric. Carbon dioxide occupies a different binding site on the hemoglobin. Carbon dioxide is more readily dissolved in deoxygenated blood, facilitating its removal from the body after the oxygen has been released to tissues undergoing metabolism. This increased affinity for carbon dioxide by the venous blood is known as the Haldane effect. Hence, blood with high carbon dioxide levels is also lower in pH, more acidic. Hemoglobin can bind protons and carbon dioxide, which causes a conformational change in the protein and facilitates the release of oxygen. Protons bind at various places on the protein, while carbon dioxide binds at the alpha amino group. Carbon dioxide binds to hemoglobin and forms carbaminohemoglobin. This decrease in hemoglobin's affinity for oxygen by the binding of carbon dioxide and acid is known as the Bohr effect. The Bohr effect favors the T state rather than the R state. Shifts the O2 saturation curve to the right. Conversely, when the carbon dioxide levels in the blood decrease, i.e., in the lung capillaries, carbon dioxide and protons are released from hemoglobin, increasing the oxygen affinity of the protein. A reduction in the total binding capacity of hemoglobin to oxygen, i.e. shifting the curve down, not just to the right, due to reduced pH is called the root effect. This is seen in bony fish. It is necessary for hemoglobin to release the oxygen that it binds, if not, there is no point in binding it. The sigmoidal curve of hemoglobin makes it efficient in binding, taking up O2 in lungs, and efficient in unloading O2 in tissues. In people acclimated to high altitudes, the concentration of 2,3-bisphosphoglycerate, 2,3-BPG, in the blood is increased, which allows these individuals to deliver a larger amount of oxygen to tissues under conditions of lower oxygen tension. This phenomenon, where molecule Y affects the binding of molecule X to a transport molecule Z, is called a heterotropic allosteric effect. Hemoglobin in organisms at high altitudes has also adapted such that it has less of an affinity for 2,3-BPG, and so the protein will be shifted more towards its R state. In its R state, hemoglobin will bind oxygen more readily, thus allowing organisms to perform the necessary metabolic processes when oxygen is present at low partial pressures. Animals other than humans use different molecules to bind to hemoglobin and change its O2 affinity under unfavorable conditions. Fish use both ADP and GTP. These bind to a phosphate pocket on the fish hemoglobin molecule, which stabilizes the tense state and therefore decreases oxygen affinity. GTP reduces hemoglobin oxygen affinity much more than ADP, which is thought to be due to an extra hydrogen bond formed that further stabilizes the tense state. Under high.
Please subscribe and thanks for watching.